Okay, so today I'm going to review some of the stuff from Lab 1. And the first two things that we will review are discovery science and hypothesis-based science. So discovery is more about just collecting data, observing, making observations in general, and you don't have like a specific question that you're after. You're essentially just going to observe, collect data, and discover. Data can be two things. It can be quantitative. So quantitative, these are measurements. These are numerical. Qualitative are descriptions. These can eventually become numerical if you make the same description over and over. But an example for qualitative, for example, for quantity would be if I tested a combination of drugs and 80% of cells died, that's qualitative. A description would be that the cells look slow, they look larger, and they're not growing. So the difference is the numerical observation that I measured this specific amount versus this generalized observation that I make. If I make a generalized observation more and more, that can become quantitative or quantitative. So essentially this uses the inductive process of reasoning where repeated observations lead to generalized conclusions. And this is what I kind of call hypothesis generating. And some of the best stuff has been made through this. For example, well, sometimes by accident even, like when penicillin dropped on a species of mold, we found out that penicillin kills bacteria. So number two, though, is hypothesis-based. And so this time you have a hypothesis, which is this tentative answer to an educated question. So hypotheses aren't just random. You've either made a discovery in discovery science already that you're basing this off of, or you kind of have like some background in this. There's some data supporting your hypothesis. So essentially you hypothesize, then you make a prediction, and this uses the if and when deductive reasoning, or if this happens, then this happens. So you're making deductive reasoning. So you're making a prediction, you're going to do a test, you're going to look at the results, and then you're going to evaluate those results. So one example would be when we tested NADH, malate, and oxalacetate, we found that our hypothesis, let's say, was that NADH is going to excite at 340. We found that oxalacetate didn't excite, malate didn't excite, but NADH did have a large observation. So if we predicted that it would start, we tested it, we got results, we evaluate, NADH was our one. So think of examples in lab that you've used these different kinds of reasoning for. I think a good discovery science one would be maybe the microscope slides with the cells. You were kind of just making observations upon the cells. You couldn't numerically make any assessments on the onion cells versus the bacteria cells, except for maybe their size. Okay, so one thing about hypotheses is that they have limits. It must be falsifiable, so it must be, can't spell, too bad. And failure to falsify it does not prove it. So falsifiable, so if you fail to falsify it, that does not prove. Okay, so failure to falsify does not equal that it's proven. So you have to prove that your hypothesis happens in a controlled setting. And a lot of times science gets away with saying that, oh, my hypothesis is true, even though they didn't account for certain variables and stuff. So for example, if I'm testing cells and I say, well, this combination works great, but I had the cells in an environment that was really bad for them. That's not a natural environment. That's not good. So my claims are not bound by just my hypothesis. Other things are getting in the way. And the other thing is that you have to make repeated observations. You can't just do it once, and this is one of the more frustrating things. You can't just see something once. You have to see it over and over and over again, at least 3-4 times before you can make that, before you can say this hypothesis did prove true. So hypotheses are very specific. They're specific questions, and often they stem from something called a theory. Theories are broad, they're general. They have a lot of evidence. So they're broad, They have a ton of evidence, and by a ton, I mean the chances of a theory in scientific terms being wrong is that of, remember the mole is like 10 to the 23rd, you know, the chances of that essentially like 1 to the 23rd or something. So, you know, when you think about things like this is like relativity, you know, that obviously works, that defines